good afternoon, and thank you very much for joining us this afternoon for a new webinar of the NewTechAqua webinar series. My name is Bernardo Basurco, and then I'm working at the CIHEAM, and CIHEAM is coordinated this series of webinar. And when I say CIHEAM, is both CIHEAM Zaragoza from my side and CIHEAM Bari. And we have with us Giada Menini. And today, together with us, we have researchers that have worked on the work package dealing with the genetics. So we have Anna Sonesson from Nofima. Hello, who is the work package leader of
Hello.
this topic. Marine Erlin from Kulmarex company. Hello, Marine.
Good afternoon.
Okay. Juan Luis Gómez Pinchetti from the University of Las Palmas of Gran Canarias.
Hello, good afternoon.
Marie Lillehammer from Nofima.
Hello.
And then we have from CISAF France, we have two, which is Romain. Sorry, which I don't see you now.
I am here. Hello, everyone.
Hello, Romain Morgensen. And Pierrick Haffrey. Hello, Pierrick. Well, he's there. Well, anyway, let's start with a brief introduction of our project. So our project NewTechAqua is a project financed under the ACE 2020 program, under a call that was named entitled Sustainable European Aquaculture 4.0 Nutrition and Breeding. Under this call, 4 projects were approved and has been implemented. Those are e-efficiency, intelligent fish feeding through integration of enabling technologies and circular principles. Aqua impact, genomic and nutritional innovation for genetically superior farm fish to improve efficiency in European aquaculture. NewTechAqua, our project, new technologies, tools, and strategies for a sustainable, resilient, and innovative European aquaculture, and future eAqua, future growth in sustainable, resilient, and climate-friendly, organic, and conventional European aquaculture. NewTechAqua, which is the acronym of the project that stands for New Technologies, Tools, and Strategies for a Sustainable, Resilient, and Innovative European Aquaculture, is a project that started in January 2020 and is finishing at the end of this year, 2023. The project is coordinated by the University of Bologna from Italy, and it comes on the participation of 25 partners from 9 countries like Belgium, Croatia, Cyprus, France, Greece, Italy, Norway, Spain, and United Kingdom. So the consortium is composed by the partners that are from the research and development, academia, from the industry, companies, either fish farming companies, aquafeed producers, biotechnology companies, and 3 partners working on exploitation and disseminations, like the Federation of European Aquaculture Producers, Green Innovate, and our center, the CIHEAM. So the strategy, what is the objective of NewTechAqua? The objective is to expand and diversify the aquaculture production of finfish, mollusks, and microalgae by developing and validating technologically advanced, resilient, and sustainable applications. The project has worked on different finfish species such as salmon, trout, seabass, and sea bream, in 4 what we call emerging new species like amberjack, migri, sole, and mullet, 2 mollusk species, oysters and mussels, and 3 microalgae species. And these species, we have worked on different disciplines like intelligence and data, production systems, improvement of the production systems and sustainability, in aquafeeds and nutrition, in the development of new products, in genetics and breeding, and in reproduction for the development and improvement of the domestication of new species. As regards the role that CIHEAM has been mainly focused on transfer of knowledge and training. And there, under this area of work, we have developed an intense program to implement 2 advanced courses, 2 training workshops, some e-learning modules, the school campaign that we are implementing at the end of the year, and the webinar series. As regards the webinar series, we have organized already 9 webinars on different topics, such as the acceptability of aquaculture, on new circular production systems, on organic or diets, functional diets, reproduction of new fin fish species, new production systems, or epidemiological models. If you have missed some of the past webinars, remember that you can watch all these past webinars in YouTube. They are already available in the YouTube channel of NewTechAqua, and you are very welcome to watch them. There is a lot of information, very interesting information about the outputs and the innovations that the project has developed. And let's start with our today's webinar. And let me pass the word to Anna Sonesson, who is going to introduce you this webinar, the objectives and the agenda that we have selected for today. So Anna, please.
Yes. Bernardo, you are just taking this, my 2 slides, sorry?
Yes, I can pass the slides for you. You tell me, and then I pass the slides.
Yeah.
Yeah, you want this one?
Yeah,
or?
yes, just
Okay.
like this. No, I just have a couple of slides. First one is talking about selective breeding in aquaculture. We know that it's a very powerful tool to genetically improve populations for different traits. They have economic importance, such as the production traits, animal welfare, also both the economic but also animal welfare, yes, and the quality traits, sexual maturations, also very often in the breeding goals. Globally, there is a low uptake of selective breeding. It's around 10 to 15%. But here we are talking Europe, and for the main species here, these industries have adopted selective breeding to different degrees. And we know that there are differences in reproductive biology, products, and the markets between the species. And we have in this project here worked on 4 very different species and industries. One of them is that we worked on microalgae. We will have 2 presentations today. Microalgae breeding has never been looked at before, as far as we know. And there's also no industry related to breeding for microalgae, although there is, of course, production for microalgae. In the Pacific oyster, we are looking at dual disease resistance. So is genetics of one disease affecting the genetics of the other disease? And it's also a very new way of looking at disease resistance. In Atlantic salmon, we have looked at feed efficiency, which is very important economically for the industries, because the feed cost makes up the majority of the cost of the production. And also it has a resource efficiency element in that trait. In seabass, we do a real feed by genetic interaction experiment to look at how different feeds affect lipid-related diseases. And here the latter one, we just got all the data, so we don't have all the results.
Mm-hmm.
But then I would like to give the floor to the real presentations. So
Right.
enjoy it.
Okay, thank you. So we are starting with the first presentation, which is going to be delivered by Juan Gómez Pinchetti from the University of Las Palmas de Gran Canaria to talk about microalgae reproduction and phenotyping for selective breeding program. So I stop sharing the screen and pass the word to Juan Luis. Okay?
Okay, I will share my screen now. Okay.
It's okay.
Okay,
go.
perfect. Okay.
Perfect.
Okay, good afternoon, everyone. I'm going to start the presentations today. Okay. And the first one is this entitled Microalgae Reproduction and Phenotyping for Selective Breeding Programs. That is one of the tasks that we have been developing in the work group 3 of the NewTechAqua project. And we have been working very hard all together with the people in from our laboratory and also from Nofima. Okay, this is me. I'm Juan Luis Gómez Pinchetti, professor at the University of Las Palmas and scientific director at the Spanish Bank of Algae, where we have been working for more than 30 years in developing the algal biotechnology in the Canary Islands. And nowadays, one of our goals is that we hold a culture collection with more than 2,000 strains of microalgae and cyanobacteria. Nowadays, also, we have seaweeds. We are starting a new program on seaweeds, but mostly we have been involved in microalgae research. Okay, just starting because we don't have too much time on the ideas that we have tried to develop in this task for the project was to go ahead with a program for microalgae reproduction and phenotyping for selective breeding, which was not very common in microalgae nowadays. We already know that we have other different approaches that I will introduce later, but talking about sexual reproduction and microalgae breeding programs is not very common from the biotechnological point of view. So we thought at the beginning that it was a crazy idea, but it was interesting for us to check what were the possibilities for developing this kind of program with microalgae. So I will talk a little bit very shortly on the ideas we try to develop, why we want to do it, how we get the approach, and how we get the ideas to go ahead with the program, and then what are the results that we get from this ideas at the laboratory, and how we can use in the future, which is something that we will have the ideas by Maria and Anna in a later presentation. Okay, the idea from the beginning was we want to have improvements on components for developing aquaculture possibilities and diets and all the stuff related with the possibilities for microalgae in this area. And as you know, one of the goals is to develop the improvement and the possibilities that we can get from microalgae and the production of omega-3 fatty acids. It was very interesting. And because we were focusing the idea on the genetics program, we thought how we can perform and how we can get the approach to improve what we get from microalgae. So this was the initial idea of the development of this strategy and task in the program. And from the beginning, we were just having around and looking around what was done with microalgae. And we already knew that the approaches already existing and tried in other programs were related with biochemical engineering and genetic modification, particularly nowadays, but also with mutagenesis. For example, we had some experience on mutagenesis approach in our laboratory, just working with strains of chlorella, for example, and some hematococcus and these kind of strains, which is working very interesting, very fine. However, we were just going through the idea of how we can get classical breeding as the same as we make with other organism species, and how we can get the genetic recombination tool to control sexual intercourses between different strains from microalgae species. Just having this idea from the beginning, we thought how we should start to develop a program like this with the small, yet very short reference that we already get from the journals. And we try to develop a program for the selection of suitable species. At the beginning, we were just looking around very common species, very common strains that are very useful in aquaculture, like chlorella or isocrasis or nanochlorosis. But we already observed that the possibilities to go ahead with a breeding program with this kind of strain was very difficult. So we were discharging some of these strains, and we were mostly focused on what the possibilities for diatoms were. And we thought that with diatoms, we can control some way the biological cycle, the life cycle of some diatoms that were already described in the literature, and the possibilities to get sexual organisms, I mean males and female cells that can go ahead with sexual reproduction was very fun at that time. So and going just in deep with the diatoms group, we fixed that probably the best candidates were the heterothallic diatoms that are described and are able to develop crossbreeding patterns between different cells that play the role for males and females, and the possibility to get recombinant progeny was very clear from the literature, and yes, and we perform at the laboratory the possibility to get single cells isolation from this kind of progeny. Well, the initial task was very difficult at the beginning because we didn't know exactly what the strains were interesting for, or were able to develop these ideas that we were organizing. And we fixed in the group of diatoms that we already have at the VIA collection in our laboratory, and we were trying different strains. Some of them were failed from the beginning, but there was, for example, one which is the Nitzschia palea that we already conserve more than 8 strains in our collection, and it seemed to us that was a very good candidate for the breeding program. But we already, after some trials to get the F1 progeny, it was already failed. But yeah, we were very lucky at that time because one of the trials that we had was obtained with Seminavis robusta, which is a very common diatom, which is has been already tried for other possibilities in this kind of approach related to the identification of sexual reproduction in diatoms. So we get the possibilities to work with different strains that were obtained from the BCCM collection in Belgium, that were having different strains, even different strains belonging to different clades that were very clearly identified, and that were able to develop this kind of male and female sexual reproduction and the breeding, the cross breeding between them. So we perform these kind of experiments with the possibilities that the collection was given to us. For the crossbreeding scheme, we use 8 different parent strains that were sexually compatible, and there were possibilities to go ahead with this kind of matting between them. And this is the table that we already performed for the crossbreeding trials. Well, it's very interesting to highlight that we have the possibility to perform matting experiments between the different both intraclades and interclades possibilities with these 8 families of parent cells that we already got. After some trials, we already get some of the sexual stages that we can identify under the microscope with the different matting experiments, and where we can observe both the pairing and gametogenesis, and after that the auxospore formation and expansion of the cells, and finally we got F1 progeny cells coming from these matting experiments. From this crossing that we already performed in the laboratory, from just starting with these 8 wild-type parents, we finally got 66 F1 recombinant progeny. It's very interesting to highlight that with this progeny, we already include in our culture collection. So nowadays we still have available some of these recombinant progeny that we get from the matting experiments. After getting these successful crossbreeding experiments, we start with the different traits that we were interested to check during the NewTechAqua project, which were mostly related with the possibilities to get new strains that were performing growth characteristics that were improved, and also the possibilities to get profiles of fatty acid composition and check if there were possibilities to get hereditability possibilities from them. So we perform studies for growth rates determination for all these 66 and the 8 starting parent materials, and we perform growth curves from all these growing cultures and trying to compare what were the characteristics for the growth performance of these strains, and we got many results. And in a resume possibility, we can check here what were the results that we got from the wild type strains and the F1 progeny that we got from the matting trials. Here you can see how more than 23 strains of the progeny that we got from the experiments were performing growth rates higher than 0.722 per day. That means that more than 30% of the new progeny that we got were performing higher growth rates in comparison with the wild type strains. In the lower figure, you can see the frequency in comparison, the maximal growth rate that we already obtained from the first 3 days data performing and for growth rates. And you can see how most of the strains that we got from the FU progeny were showing higher growth rates when compared with the control with the wild type strains that are the black bars. After that, we already go ahead with the cultures experiment just to obtain a big amount of biomass to perform other biochemical experiments. And we make in a traditional way in the laboratory, just using different volumes, bottles and so, just to get enough biomass to go ahead with the biochemical analysis. And here you can see, after getting enough biomass for that, we perform some freeze-dried material, and from this we go ahead with the fatty acid composition determination. that we already performed together with the people in the at the University of La Laguna, and we make lipid extraction and phase purification, and after that we check the concentration and the composition by GC analysis. And from these results, which were many of them, because we check all the different strains by triplicates, we have already analyzed at least and identified at least 8 strains of the progeny, of the F1 progeny, that show higher yields for high value PUFA. That means we were focusing on eicosapentaenoic acid and the docosahexaenoic acid, and also you can see in the figure the summatory, the sum of the omega-3 fatty acids. And these strains were performing already fine with the fatty acid compositions. Although at the end, we are not exactly convinced that these are a result of the crossbreeding experiments. So as you can see, we got a lot of conclusions coming from these experiments related to this kind of breeding programs using sexual reproduction, that we already know that has been less used in comparison to other breeding techniques like asexual or clonal selection. But we observed that a microalgae breeding program could show potential advantages, including generating genetic diversity and facilitating the exchange of desirable traits that we already we are very much interested to check some other of the traits that could be interesting, for example, the carotenoids content. Our approaches and results in the present study contribute to the selection of new potential species, to refine crossbreeding techniques, and develop new strategies for harnessing the potential of sexual reproduction in microalgae. It would be very desirable and very interesting to check if these techniques can work also with some other strains that could be very useful and very interesting for aquaculture, for example. We observed that these heterothallic diatoms, as is the example of Seminavis robusta, are a very good, very excellent choice for experimental control of this classical breeding approach, because we get once the breeding experiments were organized, the results were very fast and very interesting from the beginning. Yeah, growth data for Seminavis robusta indicate that selective breeding may be an effective approach for improving microalgae species, and this could be identified by this kind of traits that are very interesting for and are indicatives of the performance of this kind of species and strains. And finally, the findings on the fatty acid composition are very interesting, although well, we could not fix the idea that this fatty acid composition on the difference that we have from the fatty acid compositions should be or could be related with the heritability variation in the F1 progeny cells that we already obtained. Okay, finally, here you have the potential and the perspective for these approaches that are very new and can we would like to highlight the possibilities that we can have with this kind of approaches in the future, just checking with some other strains, and probably it would be very interesting to compare with other breeding techniques that are already working nowadays and could be very interesting. So that's all for my side. Just if you have any question, don't hesitate to write them through the question and question tool here in the Zoom tool, and you are welcome to ask for any idea you have. Thank you very much.
Okay, thank you very much, Juan Luis. As you said, we are leaving the questions and answers, the debate for the end, but those that have questions can start writing them in the questions and answers.
Okay.
So Juan Luis, okay, please. So
Yes.
we continue with microalgae, and then for that we have Marie Lillehammer from Nofima, who's going to talk about the microalgae breeding program, the experience conducted in Nofima. So Marie.
Yes, thank you. There.
Okay, we see you well on the presentation too.
That's good, yes. So I will continue a bit with what Juan was talking about and show how the results he was describing can be used in a potential microalgae breeding program. So as he also mentioned, that why we want to use selective breeding in microalgae, or the objective of this is we want to increase the production efficiency, because we think microalgae holds lots of solutions for future feed and food production, but it needs to be produced more effectively to be cost effective. And we also wanted to check if selective breeding could be used to improve the product quality. So we were diving into these two types of phenotype. One was if we could increase the growth rate, basically to increase production efficiency, and or if we could improve the fatty acid composition, so especially improving the omega-3 content of the microalgae to then get improved product quality and also increased efficiency to produce omega-3. So a classical breeding program works as a cycle. It means that we repeat this cycle a number of times and we get an accumulated genetic gain over time. So one aspect that needs to be in place is this crossbreeding, which Juan was explaining that we were able to, or they were able to perform with Seminaris robusta. And the crossbreeding stage is important to generate new variation that we're going to use for the selection later. After the crossbreeding, we need to measure something of importance. So you have already seen how we could measure growth and fatty acid composition in our F1 offspring. And this can then be used to select the highest performing strains, and then repeat the cycle with a new round of crossbreeding to get an even higher genetic gain than what we would get from one round. So this table you have already seen. We started in this experiment with 4 and 4 plus 4, so 8 wild-type strains. We performed as many crosses as we managed, both inter and intraclade crossings, and produced 66 F1 offspring. So the numbers in this table shows how many offspring cultures we produced from each crossings, showing that it varied a lot. It was easier to perform intra than interclade crossings, and some of the strains did not produce any offspring. But in the end, we obtained 66 F1 cultures, which we could go on to phenotype and measure and do this and estimate heritability and potential genetic. So as described in detail already, we measured the growth and fatty acid composition of all the 66 wild-type, 66 F1 progeny, and also the 8 wild-type strains. This graph you have also seen already about the maximum growth rate. From the dark bars here are the 8 wild-type strains, while the orange bars are the offspring. And what is important to notice in this graph is that when we do this crossbreeding exercise, we see that we increase the variation. So we do get offspring that grow faster than the fastest growing wild type strain. And that's part of the point here, that this new variation, it produces some very superior strains. And these could potentially be selected for the next round of crossbreeding again, so that we could increase growth rate for every cycle and potentially increase it a lot in the end. And I will take you through an exercise where we estimate how much could we potentially increase growth rate by this method. So we did test three different growth rates: growth over 6 days, growth over 9 days, or maximum growth rate, as I just showed in the graph. And we see that the heritability varied between 46 to 65%. That means that from the variation we see between the strains, 46 to 65% of this is caused by the difference in genetics between these strains. So approximately half of the variation is due to the genetics between the strains. And this is a very good starting point for improving this trait through selective breeding. We also tested the fatty acid composition, and we found a significant genetic contribution to the variation for saturated fatty acids and for omega-3. But the heritability was much lower, 16 to 18%. So it is heritable, it is possible to improve by selective breeding, but the rate of genetic gain will be much lower because heritability is lower. So a larger part of the variation is explained by other factors than genetics. If we assume that we put this into a breeding program where we produce a number of strains and we select the 20% best ones to produce the next generation every cycle, this would give, for the growth rates, 21 to 25% improvement in growth per generation. And the generation here is one cycle, which goes quite fast for this species comparing to, for instance, animals. So assuming that we could do 10 cycles per year, this means that we could increase growth by 6 to 9-fold in 1 year by this method. If we think of improving the fatty acid composition instead of the growth, we see that the improvement per generation per cycle is lower, it's 5 to 8%, because there are so many other factors contributing to the fatty acid composition. In addition, we could not perform that many cycles per year because the experiment needed to measure fatty acid composition takes much more time than the growth experiment, basically because we have to grow to a larger volume. So with much fewer generations per year, it means that the improvement per year would be 20–30% maybe. So we see that if we are thinking of production efficiency, it seems much more efficient to try to improve growth as rapid as possible. And then larger growth might also give more fatty acid composition, assuming that these traits don't affect each other. So we conclude from this that both growth and fatty acid composition are heritable. So they can both be improved by selective breeding, but the potential for rapid improvement is higher in growth than in fatty acid composition. So I want to include just one slide of what we don't know yet, how to move on from research to an actual breeding program. And one thing is that we started here with 8 wild type strains, and that means that our total gene pool is what is present in these 8 strains. If this were to be run for many, many cycles without running out of genetic variation, we would need to expand the number of wild type strains to start with. Secondly, we measure growth in a 12-day experiment under laboratory conditions. For this to be commercially relevant, we have to check that those growing fast under lab conditions also grow fast under upscale conditions. So that's also not yet tested. Also, if we're actually able to 6-double growth in a year, we have to test that we don't get any negative side effects of selection for viability, robustness, or biochemical composition or anything, because that's a huge change of an organism and we cannot know beforehand how it will affect the organism as a whole. So thank you. Well,
okay, thank you very much, Marie. Again, those that are interested in having questions, please start writing them in the questions and answer. And let's continue. We move now to Bible mollusk, to a presentation to be presented by Romain Morthpelsen from CISAF, France. So, Romain?
Indeed. Can you hear me?
Yes, I hear you well.
Right. So now we move on to the next presentation with a new species, moving from microalgae to what eats them, the oysters. I'm going to show the results of the NewTechAqua experiment that was on genetic improvement of survival to a dual infection in Pacific oysters, which is one of the first results in the animal kingdom, one of the first time we tested this methodology. So this was a collaborative work between IFREMER, INRAE, CIHEAM, 3 organizations in France. And I should note that most of the work was done by a PhD student, Juan, who defended this year. So first of all, why do we want to tackle the problematic of resistance to a dual infection in Pacific oysters? The worldwide production of Pacific oysters was first hit by a virus named OSHV1. It is present on the French coast since the early 1995, with initial mortality of more than 90%. So very impactful disease that puts a lot of oysters farmers out of a job. So breeding started and genetic selection was proven to improve survival. As you can show in this graph, the green bars are unselected individuals, and the pink bar are selected, and you see more yield because of more survival. That's this second graph. See here less mortality for selected individuals for this virus compared to wild spats. But then the second disease occurred from a bacteria named Vibrio osturianis, variety francensis. It was detected in the early 2000s, but started to really be a problem in the 2010s. It leads to a moderate mortality compared to OSHV1 at first, like 25%. It has been proven to be irritable, so breeding should work and does work. And there is some hypothesis of interaction between the disease, some scientific work done on immunosuppression caused by OSHV1 that triggers the bacteria infection, for example. But it's still pretty work in progress and still understanding the interaction between the pathogens. So what we wanted to tackle in NewTechAqua was to address or to look at the meaning of resistance to a second disease in context of a dual infection, because both infections occur at the same time, can occur at the same time. And how do we breed for a resistant individual when you have 2 diseases? do we have to compare the resistance independently, additively, or is there an interaction between the two? And how do we adapt breeding programs for that? So the protocol, the experimental protocol, was done by IFREMER at an IFREMER facility. They produced 100 families from 3 parental sources, one from a breeding program with a huge intensity of selection and resistance individuals, one source with wild parents from a site that with high infection pressure. So the hypothesis was that there was some natural selection occurred there, and so you would have some resistance here, and some wild parents from a site with low infectious pressure. So more susceptible because no selection should have occurred on those animals. They did a mono or dual control infections that I'm going to details right after. The genotyping for parentage assignment and genomic selection was done with cutting-edge tools, 57K SNPs, oyster B species oyster array. And we did the estimation of genetic parameters, heritability, genetic correlation, accuracy, and genetic architecture to identify potential QTLs. I'm gonna detail all of that just after. So the protocols for the disease challenge, the big innovation in NewTechAqua, it was an infection by cohabitation. Basically, you put your test oysters in contact with sick oysters with either the bacteria or the virus or both. So 3 conditions, and we know they're sick because they are injected with the bacterial or the viral suspension. Here you have some detail of the protocol, but it's pretty standard challenge. In one condition, you have 2 pathogens. The partner for genotyping was INRAE lab in France called Jean that did the genotyping for the 2,300 oysters. Some results, the mortality and genetic parameters. So here you see the mortality curve for the 3 challenges in dark blue or purple, the virus infection, in yellow, the bacteria alone, and in light blue, the dual infection. It was pretty much expected kinetics and mortality rates from what we know of those disease. So very sharp and fast mortality for the virus, and a lot more continuous mortality for the bacteria, and some kind of addition of the 2 for the dual infection. Here is the heritability and genetic correlation matrix, sorry. So heritability here in gray and genetic correlation in the green boxes. If you're not a geneticist, what you need to know about heritability, that the higher it is, the most efficient breeding will be. So if these numbers were 0, you could not do breeding. They're not 0, they are actually quite high. So there is room for genetic improvement for breeding for those diseases, in particular in the context of dual infections, because we knew heritability for bacteria and the virus, but not really for both at the same time, especially not using genomics. And in green, you see the correlation between the diseases. So also some expected results. We expected no correlation between the single pathogen challenges, and that's what we found, basically 0. And for the dual infection, we see a slight correlation with the bacterial challenge, but a strong, very strong correlation with the virus, which was the first main results of this experiment. The dual infection seems to be driven mainly by the herpes virus resistance. Then we did some accuracy of breeding values. So basically, we took our data and compared the use of genomics versus not using the genomic tools, and to see if it was worth it to genotype with the SNP array. So for the 3 challenges, in purple, just without genomics, and in light blue with genomics. And we found for OSHV1 and dual infection an improvement of accuracy of selection, and so a use of genomic and SNP array that was useful for breeding. We see no genetics for the vibrio mono challenge, bacterial challenge, but maybe this was because the sample size that we genotyped was too small. So the question is still up there if genomics is useful for vibrio. And also I should note that there is some large variation of the accuracy also because of the breeding design and the numbers genotypes. But still, we see some improvement with using genomics. So now about the identification of QTL. So we look at the genome, at the oyster genome, and to see if there was some region that was significantly associated with a resistance to a particular challenge. We see no QTL, nothing for Vibrio oysterianus here. I should explain those graphs. The 10 boxes are the 10 chromosomes of the oysters, and every dot is a SNP. And if the SNP is high enough above the threshold, it means that this region of the genome is associated with survival. And so maybe there is some gene here that could be interesting. So we found no gene in Vibrio oysterianus, but again, I mentioned sample size was an issue for this challenge. But for herpes virus and the dual infection, we found the same region on chromosome 6 that seems to be significantly associated with resistance. You have here some characterization of those region for those interested. So what are the effects of this QTL? So if we pick the genotype at those specific region of the genome, do we see actual survival rate differences between the different genotypes? We see some weak and additive effects of the 2 markers detected in OSHV1 infection, and we think there is some kind of a dominance-like effect for the markers detected in dual infections. It means that having just a resistant allele is enough to be more resistant than having both susceptible alleles. And so some conclusion and perspective. So this is the first reports of genetic analysis of disease resistance in dual infection in animals. The protocol developed has some drawbacks, one of which is the it's not the same. The experimental challenge is not the same as especially for Vibrio, as the one that we observe on field, on the farms, because the oysters used here were very, well, quite young, younger than the animal that actually get sick. And also there is some complex and multiple infection and reinfection often associated with water intake, freshwater intake on the farm that were not addressed here, but that will hopefully be addressed in the next project. So some conclusion, in the condition of the protocol applied, we see independence of resistance to the virus and the bacteria at this young age, as I mentioned. Genetic resistance in dual infection seems to be mostly driven by the resistance to the virus, which is not to say that dual infection is useless compared to herpes. It's still useful, but it is mainly driven by the herpes virus. The genomic selection seems to be not efficient on Vibrio oysterianus, but the question is still up there that maybe if we had some more samples, could have seen some progress. But it's very highly justified for herpes and dual infection. And the QTLs identified have a weak additive or non-additive effect, still to be researched further to understand what genes and what's happening on those region of the chromosome 6 of the oyster, or resistance to disease. And so that's the last conclusion. The QTLs have to be refined, but with the tool we have and the limited linkage disequilibrium that we see in the oyster genome, it will make future characterization hard, very resource demanding, but still feasible. And so with that, I thank you for your attention. And as everybody mentioned, the Q&A tool is there for you to ask the questions, and I'll try to answer them as best as I can.
Okay, thank you very much, Romain. Okay.
All right.
Please remember to start writing your questions and doubts for the debate that comes after the two following presentations. Now, after having the presentation for Microalgae on Bible mollusks, we move to fish, and then we start with sea bream, with Marine Erlen from Culmarex.
Hello,
So please,
can you hear me?
yes.
Can you see my screen?
Yes, try to speak a little bit louder if you can.
Yes. Can you hear me better now?
Yes.
Okay. Good afternoon, everybody. So I'm Marine Erlin. I'm working for Culmarex, which is part of Cook Spain company, a global company, and we are a bass and bream producer in Spain. I will present the work that we've done as part of NewTechAqua project, which was a trial that we run on some of our selected bass families to test the effect of two commercial high energy diets on growth, lipid deposition in both liver and fillet of selected families. And this work is a collaboration between Nofima and the hatchery of our group, which is called Aquaculture Balear. And the hatchery is located in Palma de Mallorca in the Balearic Island. So a little bit on the background first. As you probably all know, the aquaculture industry is under a lot of challenges, and one of which is basically try to sustainably feed a growing worldwide population. And as a consequence, the composition and the ingredients of the commercial diets are changing over time. So we need to be prepared for all these changes, and we think it's very important to take this parameter in consideration in the context of a breeding program. Also, the feed represents the main cost behind the Mediterranean aquaculture seabass market fish price. So this is also really relevant. When you buy a seabass, you need to be aware that a lot of the price is made by the feed that the fish has been consuming during its life. And there is also a lot of work that has been published over the years showing that feed composition has an impact on fish performance and product quality. So as a representative of the industry, we are very, let's say, aware of this, and this is something that we wanted to investigate in this project to be able to see if the commercial diets that we are using in our commercial production have an effect on the performances of our bass families. So this is a little bit of the context for the study. Now I have also to say that I will present some preliminary results, not much of genetics, because the trial has ended a couple of months ago, and we are at the moment heavily involved in the analysis of the genomic data and the genetics evaluation. So I apologize for that, but I will give you anyway the details of some preliminary results that we have, and also the context of the study. So the main objectives for this study were to test the effect of two commercial diets on the growth performance and the lipid deposition of the families. As I said before, what was very important for us as a company was to use diets that we are currently using for our seabass production. We didn't want to go with experimental diets. We really wanted the results to be, applied into our commercial production as soon as the project ended. And also what we wanted to do is to gain more knowledge on seasonal lipid storage dynamics, which we know are occurring during the ongoing phase at sea, because we on-grow our fish during, let's say, 15 to 18 months at sea. So they spend a long time and more than a year, so they are exposed to different seawater temperature, the natural changes over the year, and we know that all this also is giving like seasonal effects on the metabolism of the animals. And the study basically is focusing on feed by genetics interactions for the families. We used, well, I'll explain later the experimental design, but we exposed the same families to both diets, and we made an effort on having large numbers of fish to have like a representation of families to get a good estimation of this interaction. And our, let's say, main goal for us at the end is to try to find within the families that we have exposed in this challenge, try to find the families that are more resilient to the diet changes, so that are growing well, and also in terms of quality of fillet are giving good results regardless of the diet that we are using. So we deployed some innovation in this study. So for us as a company, it was the first large scale feed trial that we were carrying out. And we took the advantage of using also an experimental site to be able to on-grow those animals. So they were not on-grown in tanks, but at sea. We used new equipment and technology to record phenotypes, again, in a large scale genetic experiment context. So there was quite a lot of challenges in, getting all the phenotypes recorded, and we were a large team of people involved in those samples. And finally, for the genetic evaluation, we used the medfish array, so high density SNP array that was previously developed as part of EU project, the MED8 project, and this is the tool that we use to perform the genetic analysis, genotype our fish. And the idea is to do genetic and genomic evaluations and also study the genetic architecture of the phenotype. For phenotype recording, we use the distal fat meter to measure fat content. We also use the hyperspectral imaging to analyze pictures of livers and whole fish, and the array, as I mentioned before, to genotype the animals. So regarding the protocols, we had a setup of 6 experimental cages, 3 replicates per diet, and we used 2 commercial diets that I call Diet 1 and Diet 2. As I mentioned before, those diets are currently used in our commercial production. And you see here like an overview of the composition. So what we wanted to contrast is like 2 different diets in terms of raw material. The diet 1 had marine sources, plant sources, and also LAP, which is land animal products, while the diet 2 had only marine sources and plant sources raw material. And in terms of growth fat, you see there is a difference also in terms of growth fat content, with diet 1 at 19% and diet 2 at 24%. So as I mentioned before, we run the experiment, let's say twice on 2 groups of 20 families. So we did 2 consecutive experiments. One that was carried out during 2022 between April and December. And the second group was trialed this year between February and September. And you see here the average weight at stocking and at harvest are a bit different. For the first group, we started at 150 grams until 776 grams, while the second group was stocked at larger weight, 300 grams until 630. 3 replicate treatments per diet. And what we did also is monthly measured individuals for body weight and also fillet fat content of all the fish. and we had in total 5 measurements. And for the final sampling on both groups, we recorded the following traits. So the body weight, the carcass weight, the standard length, the fillet fat content using the distal fat meter, the liver weight, the digestive tube length. We also recorded the gender, and we also recorded the liver lipid content using both histology and the hyperspectral camera. And finally, we also took a picture of the whole animal using also the hyperspectral camera. So some preliminary results. One thing that we were really, really happy about was that basically when we studied the results from the first group, we saw that there was a significant effect of speed and gender on the final body weight, liver weight, and average fillet fat content. So we were very happy to see that actually diets were giving differences, which we were frankly a bit worried about when we started the experiment, because we didn't expect to have such a clear effect of the diet. And here are some graphs basically showing that diet 1 showed like lower body weight, higher liver weight, and lower fillet fat content, significant difference for the 2 groups. So that was a first result, which is really encouraging, because it means that actually we can exploit the data and really do a good evaluation of genetic bioenvironment study. Then we also have some preliminary results from the use of the hyperspectral camera, and again we are very happy with this. So our colleagues from Nofima have been working on the analysis of the pictures. So on the left, you see some results from the pictures of the frozen livers that was done at Nofima. And basically what you see is that using image analysis, we can also pick up differences between the 2 diets. And we see that basically diet 1, the fat percentage in the liver is lower than for diet 2. And the results seem to be very consistent. And one thing that we didn't expect at all when we started to analyze the data from the whole body pictures is that with the camera, we are able to classify the animals according to the feed that they've been eating. So this was really surprising. We are not sure about what's behind this and what kind of application, let's say at the commercial level, we can have. But that was very surprising data that actually based on this kind of image analysis, you are able to tell if the fish were eating diet 1 or diet 2. So now our colleagues in Nofima, they are working on more analysis and on those pictures, and try to find maybe some indicators that we can use. And for us, it would be, for the company, it would be very interesting, to be able to use image analysis to link that to some phenotypes, because of course, when we do chemical analysis or the expense can be quite high. So, and also the results can take quite a long time to come back. And with image analysis, we see a very good tool to be able to have almost immediate analysis of the phenotypes, and also at a very low cost. Of course, the camera is quite expensive, but then there's no cost involved with the analysis. In terms of also the evolution of the traits that we measured during the ongoing phase of the animal. So as I said, we took several measurements of standard length and whole weight and fillet fat, and we clearly saw that there was some seasonal effect on the fat fillet content, which we think is which we were expecting. And basically what you see is that the fillet fat content is increasing during the summer period, and then it is decreasing when we enter the winter period. So, and again, with the differences between the 2 diets. And what we also have been interested in is further looking at also differences between male and females, because in seabass there is a clear gender effect on growth, with the females that are growing bigger and faster than males. So we were first really happy with our experimental design, because we had a quite balanced sex ratio, which means that we can study both genders. And we had a look at the evolution of the weight per gender and given the diet. And we found something that is really interesting that we have no explanation for the moment, but we are working on the data. If you look at diet 1, so the evolution of the weight for the female diet 1 and the males, and for diet 2, you see here that for diet 1, you've got 2 peaks for the females, which means that for the last sampling, which was in December, we've got 2 weight groups, not a normal distribution, but 2 peaks. So we've got like a group, let's say, of females that have a lower body weight and others that are bigger, and which is something that we don't see with diet 2, and we don't see in both diets for the males. So we are really interested in looking further into this. We think there is certainly an underlying physiological explanation. We are not sure what it is, but we are currently working a lot on this. And well, it's really interesting for us to see that actually we have such a different response, you know, between the 2 diets that we are using, because this pattern, we don't see that on the diet 2. So what's next for us? We are, as I said, still analyzing the phenotypic information. We almost finished for the first group, but we still have to work on the second group of families. And we are performing, as we speak, the genetic evaluations on both groups of families on all the recorded traits. And of course, the end product for us would be to identify and select individuals or families which are performing well, regardless of the diet, for the traits we are interested in. So we have still quite a lot of work to do to meet the deadline of December, but we are confident we will. And of course, all the results will be later available in a report that will be publicly available. And I thank you very much for your attention, and thank you also the colleagues in Nofima and my colleagues in Spain for their hard work, and thank you for giving us the opportunity to share our results today.
Okay, thank you very much, Marine, for your very interesting presentation. I'm sure there will be questions thereafter. And let's move and continue with the final presentation to be done by Anna Sonesson about genetic potential for improved feed efficiency in Atlantic salmon. So Anna, please.
Hello, can you see the presentation? Yes. Yes, thank you. My name is Anna Sonesson. I work for Nofima in genetics. Here we worked on a very important problem, feed efficiency. It's one of the holy grails in Atlantic salmon production. And the reason is that the feed stands for 50% of the production costs. And these are quite old figures from 2019. So it's probably higher now. And it also stands for 70 to 80% of the greenhouse gas emission. It's also 2019 data. Here also you see the real definition of feed efficiency ratio that we work with here. It is the growth gain, how much fish grows, divided by how much it eats. And we wanted to know here if genetics can be used to improve feed efficiency in Atlantic salmon. And this is a pretty problematic trait to work with. in fish because we cannot get individual feed intake records, and for breeding we prefer to have individual records. So we work here with indirect selection traits. And so instead of working at the individual level, we set up a trial with 1,750 fish. You see here a photo of different tanks. We had 35 families that were duplicated, so 70 tanks with 25 fish per tank. And unfortunately, we had only rather small fish. They grew from 50 grams to 110 grams during the experimental period. Every day, the feed intake at this family level was recorded. Some results. First of all, we here could see that we had differences in feed efficiency among the families. So you have the families on the x-axis and feed efficiency. This is the wet weight. Understood well from the feed people. And here is the genetic and phenotypic correlation between traits that we work with. So we had feed efficiency here at the is one of the traits. The other one is weight gain, feed efficiency ratio, and energy gain during the experiment. And we know from just experience that weight gain, to select for weight gain, improves also feed efficiency. And feed efficiency ratio, we of course, that's the trait we're interested in. But this trait here, energy gain, it is also an indicator trait that is based on how much lipid the fish has. And if I take the results, is that we have here on the diagonal, we show the heritability for feed intake. It was only 0.35, 42 for growth, 0.19 for feed efficiency ratio, and energy gain was 0.26. So we see we have about moderate heritability for all these traits. Growth is the highest. Here we had individual records. The other ones we had only the family records. But what we were very happy to see is that the relationship between weight gain and feed efficiency was 0.7. So it means that we could actually quantify the effect of growth on feed efficiency. But we also see that this energy gain was negative, but equally strong relationship with the feed efficiency ratio. So with this, we conclude that if we could, but the correlation between feed efficiency ratio and energy gain is quite low. 0.2. And it's this one here. It means that if we can then include both of these traits in the breeding goal, we would improve the selection for feed efficiency ratio more than we do when we only have growth indicator. For people here that are interested in nutrition, this shows that there is a relationship between feed efficiency ratio and protein retention. The R-square was 0.86. So it's, to improve feed efficiency is important for resource efficiency, since protein is a scarce product. My colleague did an economic calculation on this data. And we were especially interested in this energy, of this lipid that was on the fish. So typically a slaughter-sized salmon contains 21 to 23% lipid. And he calculated that if we could reduce 1% unit reduction in body fat, that would result in 3.3% reduction in energy content of the fish. And that results also in 1.65, so about 50% with 3.3 is a reduction in the feed used, given the same fish size and growth. And because we have a large production of salmon in Norway, it ends up with a large economic effect, up to 40 million euro reduction in annual feed costs for the Norwegian salmon production. And we also calculated that it would take around 3 generations to reduce by 1%. But still, it's a lot to gain here. So the conclusions is that body lipid content and growth, they are good candidates for indirect selection to improve feed efficiency ratio in Atlantic salmon. Improve feed efficiency ratio says protein. And we would like to validate these results in the large fish because it's the period out in the sea that they set on the most fat, the most energy. Here are the implication for the movie company that we got the fish from. This text is from Ashley Norris, and she said that the feed efficiency trial demonstrated the extent of genetic variation for feed conversion ratio and daily feed intake and the potential to improve this trait. If there was a feasible way to phenotype it, energy and fat is a candidate to go forward with addition to growth. So luckily she can do it as we. And thank you to all these people from Nofima and Mobi. Thank you very much.
Okay, thank you very much, Anna. So I think that thank you very much to all the speakers. So it's time for the attendance to benefit of this time that we have for a debate, for the questions and answers. Maybe we can start with the questions that have been already written. Well, there was one not very related with this, very much related with this webinar, which is about the use of insect meals in European aquafeeds. And Anna was providing a link about the legislation that is in Europe, which is say that anyone using insect meals may have the same legal requirements that for any other ingredient. So this is some way related with some of the issues that have been presented here, as in microalgae selection, trying to look for microalgae that have higher fatty acid contents in order to be able to provide the industry with other alternative sources of fatty acids, which is probably the fish oil is probably one of the most scarce ingredients that the industry has, and that impacts very much on the prices. So we have seen the three or four presentations about this problem, no? About the need to find new sources of fatty acids and also to use them properly in order to avoid fat deposition in the body or in the lipid. So, well, I don't know, Anna, if you would like to answer. Yes, because there is a question about the pathogenicity of the vibrios estuaries concern. I don't know if Romain, you would like to comment that question?
I have responded.
You have already responded
by text by providing a link for a publication using this protocol if anyone is interested. And please do be in touch if you have more questions after that.
Yes, well, one question for you, Romain. I mean, you have made these dual infections experiments showing that is possible to make breeding selection against dual infections.
Yes.
which I think which is very, very, very interesting, because together with well, feed cost, mortality is probably one of the main constraints in production.
Mhm.
And for
Yes.
certain diseases, there are no tools available.
Uh-huh.
So, I mean, you have explained in your presentation that selection is better against the herpes virus than against vibriosis.
Uh-huh.
But if I well understood, what you are recommending is to make selection against dual infections,
Yes.
even though one of the diseases is not so well selected, no, that the other.
Yes. So there is differences between the virus and the vibrio challenge genetic parameters. But I didn't show all the results, but one of the other results was the estimation of genetic gain using one challenge another or the other, and to improve overall survival regardless of the infection, be it only virus, only bacteria, or both at the same time, you should select on dual infection. This is the challenge that allows for the most genetic gain for the three possible diseases. So I think that was also one of the unique and new results that NewTechAqua that was found in the NewTechAqua project, that dual infection can also improve resistance in single pathogen infection. Yes.
Just one question. When you have worked with strains that have been already selected for herpes virus?
Mm-hmm. One of the strain was actually selected for herpes virus for 5 or 6 generation, but also for vibrio resistance in 2 or 3 generations. That was part of the parental origins that we used to make the families.
Mm-hmm.
Yes. And that was an experimental line from IFREMER facility, and that was it was mass selection just on single pathogen evaluations.
Mm-hmm. Okay, thank you. So I don't know if there are more questions about you. I don't think so. Okay, if not, we can move to questions about the other issues. Like there is one question for Marine Erlen that he has already answered. Is about the one parameter that it was about the density. So you have already answered about this. Mm-hmm. Okay. Yes, I don't know if Marine, if you can tell us a little bit more about the difference between the two diets that you have used in your experiments.
Well, basically not much more than than what I said in the presentations, because those are commercial diets, and we don't really want to disclose the raw materials. No, just to say that they are commercial, and really the difference that we were focusing on was the amount of fat, let's say. They are called both high energy diets, so we supply them to our seabass during the summer to promote growth. And so there was a difference. One was had a 24% of fat, and the other one 19. In terms of energy, they were more or less similar. In terms of protein content also, but there was differences in the raw materials. So one had some land animal products raw materials, which the other one didn't have, and they both had like marine plant-based products. That's as much as I can say.
Okay. Also, you have mentioned that you found interesting results in the use of those diets related with seasonality. So in that maybe there is no need to feed with high energy diets during the summer.
Yeah, well, yes, that's what we want to see, and probably work on the window, the most adequate window also for feeding those high energy diets. to comply with optimal growth yes, and not maybe having fish that are so too much fat, especially the fat that is in the gut, for example, that at the end is not efficient for us if we do filleting, for example. So that's one thing that we want to have a look at. And the other thing is also what's happening between the males and females. What are the dynamics, that are taking place during the winter and the puberty, for example? That's one question that we have also with like the two peaks of the females that I showed in terms of weight, the two groups. We were wondering if one of them was clearly indicating, well, animals that were entering in puberty. So that we're slowing their growth and devoting some energy to first puberty. So that's also, of course, a problem for us in terms of farming. Puberty, maturation, early maturation is a big issue too.
Mhm.
So I didn't want to put that in the presentation because, as I say, we are still working on that and we are not sure. But this is one of the hypotheses that we are considering. So if this is the case, if we have like a diet that is, promoting, for example, early puberty in females, this is counterproductive for us. Having those females that during the winter are devoting energy to reproduction or starting to put in place the first gonad. Although they don't fully mature, there's a slow in growth because they are diverting. Putting in place the production. So,
Okay.
yeah.
One more question is, thanks. I wonder if thanks to this research that you have conducted in cooperation with Nofima,
Yeah.
in the one you have used different ways to study fat deposition, like this fatimeter or the imaging methodology you have used. If you are thinking on using this not only in research, maybe
Yes.
in production.
Yes. So for the distal fat meter, we already use it in our breeding program because two things that we really like is being able to measure phenotypes on live animals. So we can measure that on our booster candidates, for example. So the distal fat meter is very useful because you don't have to kill the animals. You can anesthetize them and take measurements without, it's not invasive. And in our previous work that we did with Anna and her team on the Medate project, we showed that there was a good genetic correlation between the fat measured in fillet and the distal fat meter. So that's really encouraging. The distal fat is not measuring fat as per se, it's an indirect measurement. So in absolute values, when you do chemical analysis and measure or get the measurements in the distal fat, it's not the same. But genetically, they are correlated, which is what we are interested in, and that we are selecting the same traits now. And then the other thing is that we are very interested in the hyperspectral camera. For us, it's the first time that we have access to this technology and can implement it in our sampling. And we think this is also the future, and the fact that we can take a picture that's, okay, the camera is an investment. But then, we can get a lot of information from imaging analysis, and same thing, we could do that on live animals as well. So that's something that we are very, very interested in working on those fields.
Okay, thank you very much.
Probably.
Okay. I don't know if Anna, you would like to comment on something, on clarify on some of the questions.
No, I don't think so.
No. Well, so maybe I have one question.
At least I can say one thing, and it is that
Yes.
I'm happy that at least in the 3 big trials here, I think we had an okay power to perform the experiments. The feed efficiency with Gómez, we had very good power. I think you had quite okay power in your experiment, Roman, didn't you? Yeah, and the same in the Atlantic salmon.
Yes, indeed.
I think that's, I'm very happy about that. It makes the experiments expensive, But at least we then also have a higher chance of finding results.
Yeah.
And for the microalgae, of course, this is the first time we worked on it. We would have wished to have more, but at least we found good results to work on with.
Yeah. Well, actually, I found very interesting the results on microalgae. Very interesting to see how much with a little selection you can improve so much the growth. Maybe not so much the improvement on fat content, no, but then I imagine that looking at this, no, being this the first time that breeding selection is done with microalgae, these results must be very exciting for those of you that are working with microalgae, no? I suppose that one of the recommendations, for, at least for discussion, is how you can use breeding selection in combination with other methodologies that you are already in use, as biotechnology or production systems to improve the efficiency of the fat content, for example.
Yes, well, I will make a comment on that, if it's fine. Yeah, it is very interesting, Bernardo. I must say that I am very much surprised about the results. We already got it, because at the beginning, you know, that we were very, very afraid that we could not get trustable results. But I think there is a lot of work to do, because it is very interesting. Just look for other strains that could be also very interesting for aquaculture and for biotechnological purposes, because this is a very new approach. Nowadays, there are many people just working on genetic modification, and also mutagenesis is a very interesting tool. But for example, with mutagenesis, the amount of success or percentage of success after n generation is very low, because mutagenesis sometimes is reversible. So my discussion and my concern about these possibilities that we can go ahead with new trials. We still have in the collection, we have conserved the generation that we already got from the experiments, and we can try again if we have the possibility. And even we are just searching for new strains to check these techniques, right? So I think it's very interesting, and we will try to go ahead with all these methodologies and ideas.
Good. I don't know if well, Anna, you would like to say something, no?
Just a comment to this. I think, Maria, you also have to go in here, but this species or groups of species that we work with here, the diatoms, they have a phase where they have sexual reproduction, and that's not for all microalgae. And there, the breeding program would probably look more like a plant breeding program, or where you have, so I think this is just a start for a specific type of microalgae. It's the whole universe in its own, as understood.
Yeah, no, and if I may say something, is very nice, because the legal framework in Europe is against the genetic modified organisms, at least for the moment being. So seeing that with breeding programs we can improve the same or better, no, that we can be obtaining in other ways is very positive, because it gives us more possibilities to respond to the demand of the industry, no, because fish oil is very scarce and is impacting very much on the prices, no, and on the availability of affordable fish diets, no. I don't know if Marie, you would like to add something or to what has mentioned here.
Yes, no, I can just agree with the other two that this crossbreeding phase might be the most complex for many species, as I've understood, and it was not straightforward, as I understood, in this species either. There was lots of attempt, and not all combinations of strains were possible. So that's a very important
And a
area.
huge effort by Francesco.
Yes,
must be said.
absolutely.
Yeah, you're right.
And so they worked
Maybe we
very
should have
hard
been here.
to make it happen. And for some, for lots of species, it would be possible at all, and for other species, it's a huge job needed to manage and control this crossbreeding. It's not enough that they can reproduce sexually, because you also have to be able to control who mates with who among these strains, which is a very important and complex phase of this procedure.
Okay, thank you. Well, I don't see more questions. I don't know. I wonder if Pierrick Haffrey would like to say something. Eh?
Not really. Thank you, Bernardo. I think also as Anna that in the project, a very good job was done on very, very extreme or different topics according to species. It was very nice. I have just a question, Marie, about the results you present about the difference between the parents and their progenies or their families produce. How do you explain that the progenies may have higher level of growth, for example, genetically speaking, if
Yes,
there is
so
no selection of the parents?
no, what we have done is we did on purpose some crosses to contrast also, because we wanted to have some differences in terms of in terms of phenotype. So we have a nucleus where we keep well, we have kept also different the types of phenotypes and contrasted families. So we use those for those kind of experiments to try to get a good grasp of the diversity of phenotypes we can get.
Excuse me. My question was more about Lillehammer. But I understand your response.
Yes, so you wonder about the
sorry.
distribution of growth, why some of the offspring has higher growth than the best parent. Yeah, it's because that's actually expected when you cross existing strains, you recombine genes. I'm not talking about physical recombinations, but just that the parents' genotypes are combined in a new way, because you create a new strain that is half one parent and half the other parent. So actually in an ongoing population, half the genetic variance is regenerated this way every time you perform the crossbreeding. And that's why this stage is so important in the selection scheme. So it means that when you cross the parents, you expect that you will have high variance among the offspring, given that the parents are not related to each other. So that's why you need a large number of parents if you're going to scale up this. And that means that some of the offspring will be better than the best parent, while some will be worse than the worst parent. And that gives the variation you need to perform selection in the next cycle.
Thank you. Very pedagogic.
Thank you. Okay, I don't know, Anna, if you would like to add something more.
Well,
No.
I think
Okay.
we're fine.
Okay. Let me see, because I was trying to show, because I think that we can end here, but before ending and closing the webinar, just to remind our attendees that they can watch past webinars on our YouTube channel. Okay? So we have been recording the webinar, and in a few days this webinar will be uploaded in the channel. Okay? So thank you very much for all of you for attending with so much interest this webinar. Well, we finish here, Anna. So
Thank you, everybody.
thank you to all of you.
Thank you very much.
Yeah.
Thank
See
you,
you
everybody.
soon. Bye.
Bye.
Okay.
Bye.
Cheers. Bye bye.
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 Cheers.